Hello, myself Dr. Sayantani Sihi Arora. I'm the Chief Scientific Officer at Agrivet Research and Advisory Private Limited. We are basically a contractual research organization and we are situated in Kolkata, West Bengal.
What can you let us know about the shift to molecular diagnostics compared to traditional methods?
Okay. So first I would like to, in that case, I would like to introduce that since I am a molecular biologist, I did my PhD in molecular biology. So bringing that knowledge to poultry industry, it was like what we faced or what we came across. It was like it was for the disease diagnostics. Like what we are using, the conventional methods that we are using, the conventional methods like we are getting some symptoms and then the vet who is going over there and they are looking for the diseases, then the postmortem is being done, they are further moving for the diagnostics, so it is taking a lot of time. So what we suggest or how molecular biology is taking over there is that whenever you are getting some sort of symptoms, the most easiest thing is you just get the samples and you can test it. So you will get a precise diagnosis or you will get what the issue is. Instead of searching over here and there, putting medicines or putting vaccines, you can directly pinpoint what the disease is. So this is how, using the modern techniques like the PCR, you can directly move towards the pinpoint of the disease. You can identify the disease and you can further move towards it.
What is the feedback of the clients when they see your service and the, the advancements, the solutions that you can offer through this kind of technology?
Well, the feedback from the customers are it is great. Like those who are understanding it, they are sending more and more samples for the molecular diagnostics, because first of all the samples, the like what happens is mainly the poultry areas, they are in the outskirts and the farms and all, so bringing samples from there to lab is a big problem. However, for molecular biology, like there are FTA cards which are making the sample transport quite easy. So in that case, just putting it is like sending letters to someone. So you can send the samples. So that's why they're liking it, because they're getting the results and it is beforehand they're getting the results. The time consumption is quite less. We are using molecular biology, the reports they are getting, they are quite fast. And moreover, in addition to it, like there is another thing, that is the DIVA PCR. DIVA PCR is for differentiating between the vaccine and the field strains. So like the birds, mainly it is for MGMS, we are doing it presently. So suppose you are getting symptoms, you are getting the MG positive, whether it is for my field strain or my vaccinated strain. So those differentiations can also be done. So definitely these types of monitorings, regular monitoring, can help the birds, and it can be a great boon to the farmer, whosoever is using it.
What is the biggest change or challenge you have noticed in the poultry industry recently regarding your field?
The biggest challenge is definitely, I would say that because it is not known to many of the people who are there in the poultry industry, what they normally suggest is they ask for basic tests. Like they go for the postmortems and then how they can they go for ELISA. However, what I would suggest is that with using ELISA, definitely if you have your vaccine history, you can monitoring, serology monitoring is good, but you have to move a bit forward for identifying the diseases. And another thing is, since like in molecular biology, what is there is that the starting material, it is if there is a little bit of infection in your field which might not be visible but it is there, so that can also be identified using these techniques. So that is what sometimes I face, like the client is telling that we got this type of test positive, but till now I am not getting any such symptoms. But what we suggest is look for it, you prevent for it, because even if it is little in amount, it can diagnose molecular disease. These techniques, they can diagnose. However, normal traditional techniques, when the symptom is too much, at that time only it can diagnose. So these are few of the problems which I think will be taken over time by time.